so much. Can you guys hear me? This is the kindest introduction. So thank you so much. I think we've met in a conference many years ago, and you've been visited us at Stanford a couple of times, so that was great to have you around. So I'm very excited to be here. This is the first time here at Neurospin, and I've just seen a tour of your really magnificent institution, so it's really remarkable. I'm going to talk about a snapshot of my work. So this is very recent work coming out of my lab. It is going to be about vision. It's going to be rather computational. The first two authors on there's a couple of studies are Eshed Margalit and Don Finzi, who just graduated in the last 1 or 2 years from my lab. And this is going to be in collaboration with Dan Yammans, who's a professor in our Neuroscience Institute, and Kendrick Kay, that I've been collaborating with for over a decade. And just before I go into like the most recent stuff, I want to give you a little bit of history from the 90s of how I even got here in the first place. So I actually studied engineering as an undergraduate in Israel, but I like to read a bunch of stuff. And one day I encountered this paper in Scientific American. I knew nothing about the brain by Samir Zeki, and then in this paper he was describing in very lame person terms what happens in the brain when you look at the world. For example, this girl, she's looking at this art, and then he had the schematic which really caught my attention and really got me fascinated in the topic of vision. So basically said, like, vision is really rich. It's a very complex input, has many components, and it's like a really complicated task. So what the brain does when you process vision is breaks it down into smaller tasks that can be done in parallel and are much more simpler. So, and then he describes the layout of the visual system, that there's like multiple layers, like V1, V2, maybe V4, V3, V5. And within each of these stages, there are also different neurons or cells that do different computations. For example, if you want separate cells, compute colors and edges. And as you go up this kind of processing stream, you get higher order intermediate areas that do things like color vision or motion. And I knew nothing about vision, but I thought, oh my God, this is so cool. I have to figure out how to do that. So I applied for a master's and then a PhD in computer vision, actually, in the Weizmann Institute of Science, because I came from engineering. And then I worked with Shimon Edelman and Rafi Malach and started to do computational models of the brain, and then eventually fMRI. So this is still a topic that is really of high interest to me, because as I'll show you, we've learned a lot since the 90s. But I'm really interested, what's really fascinating to me about this picture is that the brain is super organized, both functionally and spatially. And one of the things that I'm really interested in is understanding why. Why is the brain organized in such a manner, and what function might it serve? And another idea that came from the 90s, this is like a more scientific paper that I read at some point during grad school. It's a paper from David Benessen and Jack Kalanit, who is in Berkeley. And they kind of had taken like the Zeki idea for a step further and said there's maybe a sequence of stages called a hierarchy, and different processing streams or different hierarchies might be optimized for different behaviors, such as maybe the ventral stream for form and the dorsal stream for spatial functions. But of course, a lot of things have changed since the 90s, and especially with fMRI, we've really been able to move from the monkey research to the human research and carve the space of the visual system. So first of all, in Samir Zeki, there are 5 visual areas, V1, V2, V3, V4, and V5. At this point in time, we know that there are more than 3 dozen visual areas in their brain, and they span actually 3 processing streams. They all start in V1, V2, and V3, which I'm going to refer to as early visual cortex. Then there's one processing stream that goes down from early visual cortex in the occipital lobe to the inferior ventral aspects of the temporal lobe, and we're going to call it the ventral stream. And then there's another stream that goes from V1, V2, and V3 up to the parietal lobe, I'm going to call it the dorsal stream. And then there's a search stream that goes from V1 to MT to the superior part of the superior temporal sulcus, and that's going to be the lateral stream. One feature of processing streams is as you ascend this processing stream, the basic computational unit, which is the receptive fields, increases across the hierarchy. So this is a picture of Kendrick. So you can imagine what happens in your brain when you're fixating in the middle of Kendrick's space. And I'm showing you a receptive field 2 degrees off the center in different visual areas. So you can see that in V1, the receptive fields are very small and local. They can maybe process little features, local features like edges. But by the time you ascend the hierarchy, maybe going into the fusiform gyrus, where you see face-selective regions, the receptive fields are so big that they might encompass a face that is in a talking distance for you. So one feature of this hierarchy sought by researchers that higher level of these hierarchies process more global aspects of the stimulus that are more tied to perception and behavior. Another feature of the visual system of the hierarchy is not only the spatial window increases across the hierarchy, but also the temporal window. So on the y-axis, you can see the spatial receptive field size, and on the x-axis, you can see the temporal window of integration. And you can see that as you ascend all 3 processing streams, receptive fields become both bigger spatially and process over a longer period of time. So if a V1 receptive field is about on average ½ a degree and it has a temporal window of 60 milliseconds, by the time that you get to the ventral occipital region in the ventral stream, the receptive fields are on average 5 times bigger, and they process things over maybe 100, 120 milliseconds. And most importantly, we have better computational tools that, such as deep neural networks, that really are really good at, they're inspired by this hierarchical architecture of the brain. They perform local computations. I'm going to talk about convolutional neural networks. And this combination of linear and nonlinear operation turns out to be really good at predicting brain responses. So now we have the tools to start asking real questions about the brain and to predict them using deep neural networks. So the roadmap for today is I'm going to take all this information together and ask 2 big picture questions. The first question, I'm going to focus on the ventral stream, and this is a processing stream again that starts from V1 and goes into the ventral temporal cortex, is associated with visual recognition and categorization. And then the second part, I'm going to talk about this kind of organization, this large-scale organization into streams. I'm going to ask why my basic brain organizes into streams in the first place. So I told you a little bit about this kind of idea of a processing stream. And then I want to talk about 2 other things. One is what kind of features might be represented in different parts of these processing streams. And second, another really big feature of the brain, and especially of the visual systems, is that there are maps everywhere in the visual system. So for example, this is a lot of work that's done in electrophysiology, especially from the days of Hubel and Wiesel. The research have found that neurons in V1 are tuned to specific low-level features. For example, different neurons are tuned to different orientations of edges. Other neurons might be tuned to the spatial frequency of the stimulus. And then there are these, you can see these dark patches. These are patches that are tuned to colors, and they kind of form like kind of a grid-like structure in V1. And the other thing about these features is that neurons with these features are not scattered randomly across the cortical surface. For example, orientation maps kind of are organized really, really smoothly across the brain. So if you move across the brain, you can see a really nice and graded topographic gradation of orientation. The same thing about spatial frequency. You can see that there is an organization of spatial frequency across the brain. So we call this as a scientific term a topography because there is a spatial map across the cortical surface of a specific dimension of visual coding. So this is true about early visual cortex, but if you you also look at the end stages of the ventral stream, if you look at the ventral temporal cortex, you can see that there are also maps. These maps span a little bit bigger in region. And here you will see maps of categories. So you can see these are activation maps across ventral temporal surface for different kinds of objects like faces or cars and houses. And you can see that there's these distinct patterns of activation across ventral temporal cortex for these different object categories. And these maps are very consistent within and across individuals. So if you show a different face or a different house, you'll get a very similar activation pattern. So you can classify from the brain responses, just looking at these activation patterns, what is that category that a participant is looking at. Some of these categories, especially ecological relevant categories, as faces, body parts, words, places, now food, have clustered regions within ventral temporal cortex that really respond very strongly to the category of interest. And these clusters that I'm showing you, just to give you a sense of scale, they're about 1 centimeter in diameter. So you can see that there is a really structured organization and structured topography, both in the submillimeter scale. So basically the orientation columns in macaques are about 300 microns in diameter, as well as categories that span about 1 centimeter in diameter. And one of the insights from this work, from this deep neural network, such as the work that was done by Dan Yammans and Jim DiCarlo, has shown one insight is that networks that are trained on a particular behavior seem to be really good at predicting neural activation. So this is a picture from their PNA's paper, where they trained many, many different networks. So each dot here is a network. And you can see the x-axis is showing how well it does at categorizing the object in the scene. And the y-axis shows how well it predicts neural responses in macaque IT, which would be the homology of human VTC. And you can see that the better it does on the task, like object categorization, the better it predicts neural data. So this has been a real breakthrough paper, because it suggests that training neural networks on the task of that network will really be good, not only behavior, but also of predicting neural responses. And this has really brought an explosion of work in neuroscience, and especially visual neuroscience, because deep neural networks not only can transform any function, any input to any function, but they can also seem to be really, really good at predicting the functional responses of the primate brain. However, one problem with these deep neural networks is that they really are good at predicting the function, but then they don't really have any spatial structure. So they can't really predict why there are maps of features in the brain. And when I personally look at maps like this, it's not only aesthetically pleasing, it really makes me think, because to get a system to be really organized require a lot more work than having it not organized. And it doesn't seem to have a big impact on the computation that emerges from a system. So the real question to me as a neuroscientist is, why are these maps everywhere in the brain, and what kind of constraints is the biology imposes that generates these maps in the first place? So because these deep neural networks don't really have any spatial structure, we had to invent a new kind of deep neural network that has some spatial organization. And the goal of our project is to understand what kind of constraints and what kind of networks can replicate the topography of the brain. So I'm going to call this a topographical deep artificial neural network, or TDANN. So if I use TDANN, it's going to be what I'm referring to. And this topographical deep neural network is based on a standard convolutional neural network. We use in this paper a network called ResNet 18. And we chose this network because, one, it's really good at predicting neural responses, and two, because it has similar number of stages as a human visual system. So one of the differences between there are two differences between the standard deep neural network and the TDANN. So the first thing is that each layer, each convolutional layer in the TDANN has, each unit is actually assigned on a simulated cortical sheet. So we have simulated cortical sheets assigned to each layer. The fourth layer of our network is a good layer that predicts V1 responses. And the last convolutional layer of this network is a good network that produces VTC responses. So basically, if you look at the square in the visual image, this is how it's going to be actually represented as you ascend this topographic neural network. So the assignment of the units is assigned prior to training, and they're just assigned retinotopolically because that's how the visual system is organized. The other thing that our deep neural network is different is that when we train the network, usually you train it to do a task, let's say object categorization. This is kind of like a task loss function. In our network, we introduce another component, and that's a spatial loss function. And the reason that we were inspired to do this is because one of the things that biology might need to do that a computer doesn't need to do is actually pack the brain into the skull. And also, different than, I don't know, OpenAI, who has infinite resources, we survive in something like 2,000 calories a day. So we have to have an energy efficient system and also a physically constrained system. And one of the principles that has emerged from the neuroscience literature is maybe the biological systems strive for a solution that minimizes spatial loss or tries to minimize the wiring lengths of neuron. So basically, when we train, we're going to train a network that has two components. It has a spatial loss component, and it has a task component. And you can control how much you want to weight these different components with a parameter called alpha. So alpha equals 0 means that there's no spatial loss. And basically, you're going back to like just a task only or standard vanilla deep neural network. Of course, also we can train with different kinds of loss functions, and I'm going to show you how that might matter for the evolution of the topography of the brain. So I'm going to show you results with the network that works best. So this network is a self-supervised network, meaning that it doesn't really have feedback during training. It's based on an algorithm called SimClear. And what this algorithm does is it takes two instances of an image as an input and asks the network to kind of map it at the end stage to be the same item. So it's basically a very simple self-organizing rule. We have a relative loss function that basically encourages neurons that are units that are spatially close on the simulated cortical sheet to have similar responses during training. So basically that's the spatial loss. And I'm going to show you results with alpha equals 0.25, which is an intermediate spatial weight. We train the images with ImageNet. ImageNet is really good for training for the visual system. It has millions of images from many categories. And again, the fourth layer in the network is the most V1-like, so I'm going to show you results from this one. And the last convolutional network is the most similar to the VTC, and I'm going to show you results from this one as well. Okay, so the first thing that we did is after training, we want to use independent data to evaluate both the spatial and the functional properties of the neurons. So after training with ImageNet, we have created another similar set to see if we can replicate different features of our network. So for example, we know from V1 that different neurons are tuned to different orientations, different spatial frequencies, and different colors. So we generated similar sets and we wanted to see what is kind of tuning that emerges in the most V1-like layer that we tested based on the ImageNet images. So you can see that neurons in the units in the V1-like layer developed tuning to orientations, and different units have different preferred orientations and different widths of orientation tuning. Also different units in the V1-like layer also develop different tuning to spatial frequency. So we can see that we can replicate functionally the predicted properties of V1. But how does it look like in terms of the topography? So we also see that the V1 This is the orientation map and our simulated sheet of V1, and you can see that there is smooth tuning to orientation that very much resembles what we see empirically in electrophysiology data. So I'm going to zoom on kind of little sections of V1 and start to see how we can quantitatively evaluate how good our network actually predicts the responses of V1. Because these orientation maps are really small in spatial frequency, we're going to use primate V1 data, which has higher resolution than human fMRI. So I'm going to concentrate on little regions like this and compare quantitatively data from the macaque. So the macaque data for the orientation and spatial frequency is going to be shown on the left, and this is a paper from Ian Nauhaus and colleagues that was published in 2012. And for color blobs, we took some data from Marge Livingston and David Hubel in 1984 that showed that there are clusters of neurons in V1 that respond to color. And you can see these are the emerging maps that come in our TDANN to these features. So again, you can see these, you get these clusters of units through the different orientations. They move smoothly, and they kind of seem to be similar in size to what we see visually in V1. So to quantitatively compare the maps, we calculated many aspects of the feature. You can see many properties in our paper. I'm just going to choose just one of them here for illustration. So one of the things that we calculated is how smooth these maps are, because as I showed you, these topographies are kind of continuous on the cortical surface. And so the macaque data is going to be shown here in the green line for each of these features. And our hope is that our network kind of reaches the smoothness of the maps of the macaque. So it's not under it or over it, basically. So you can see for all of these dimensions, the smoothness of the TDANN is very similar to the macaque. It's not statistically significant. You can also wonder, maybe it's just enough to train it on categorization, because a lot of papers have really shown that that's a really good task for the ventral stream and doesn't really need the spatial constraint for these maps to emerge. So this is the same training task. This is a self-supervised test, but we had no spatial weighting, so it's kind of like a standard deep neural network. And you can see that we get neurons that that develop the features of orientation, of spatial frequency, and of color selectivity. But you can see that these maps are much less smooth than the maps that evolve when we put a spatial constraint. And if you don't train a network, you just have this like initialization. Just the fact that we've initialized the network retinotopically actually doesn't really train. It doesn't have units that are tuned to the features, and this map has no smoothness whatsoever. So the retinotopic initialization is not what is generating these maps. For V1, there's a big literature of self-organizing maps, and basically self-organizing maps are maps where the features are predetermined, and it's trying to find the optimal arrangements of neurons on a cortical sheet. And we wanted to see how our algorithms compare to self-organized maps. So we tested two kinds of self-organized maps. The one that's called SOM is just basically bespoke self-organized map that we have handcrafted the features, kind of like traditional neuroscience research. And the DNN SOM is basically, some people have developed SOMs for V1. So we use deep neural networks to kind of generate self-organized maps. And you can see that especially the bespoke maps have the right features and they kind of look really smooth. But you can also see that these maps look way smoother than the macaque data. And again, if we quantify this, it overshoots compared to the TDANN. So we're doing pretty well in V1. How are we doing for high-level visual cortex? Again, for high-level visual cortex, the feature of the brain is that it has these representations for categories, and I'm going to show you again one feature is this, are we developing categorical information in our TDANN? So for this, we use the data from Kendrick Pay. It's called the natural scene data set, and one of the things that it has in addition to the natural scene data set is responses to 1,440 images from 5 categories. These are all grayscale images. This is the functional localizers that we've developed. and this is high resolution 0.8 millimeter data from 8 individuals. So what we can do is we can look at the categorical structure that emerges in VTC, in the human brain, and compare it to the categorical structures that emerges in the TDANN in the large last convolutional area. So I'm showing you the representational space in the form of a representational similarity matrix. And you can see that the human brain has very distinct features. For example, animate categories like faces and bodies tend to have similar representations, shown in the kind of pink and red. And inanimate categories like cars and houses also have similar representations, but the animate and inanimate are really different from each other, as shown in the blue colors. And you can see that this animacy is also observed in the TDANNs, that representation of faces and bodies are more similar to each other, let's say, than faces in cars or faces in scenes. So the representational similarity, you can see, again, this is the human-to-human data in the green, and this is the TDANN, and you can see that the TDANN really generates very similar representations. It is interesting that if we look at VTC, when we train it on the self-supervised task without any spatial constraint, we can actually also generate the same representational structure. You can see it's approaching this human-to-human ceiling. But if we don't train the network, it actually doesn't have any kind of categorical structure at VTC. But what about the topography? And if we also tested two self-organizing maps, the DNN SOM, and this is a paper from Merlin Berman's lab. It's called the ITN, Iterative Topographic Networks, that explicitly minimizes line lengths. And you can see that it doesn't really generate the same representational structure functionally. So how about the topography? So if we look at the topography, we do have units that develop features for different categories. And what's really interesting is that these features are, again, not organized randomly at this high-level regions, but actually form clusters. And you can see these red clusters are clusters of units that seem to be selected for faces. The green ones are clusters of units that are selected for places. We even get clusters of units standing like this for characters. And it's interesting because actually ImageNet doesn't really have words in that in an explicit way. It's not really a category that we're training here. The network doesn't really know how to read. And then we also get some clusters for body parts. So this is the TDANN, so we can get this kind of clustering, which is really cool. What about the TDANN without a spatial constraint? So again, we calculate the smoothness, and this is very similar to the smoothness of the human brain. If we take the task-only network, it does generate a very similar representational space. It does generate units in the high-level simulated cortical sheets that are face selective or place selective or body selective. It does develop these units, but you can see it has no spatial structure. It really does not develop the topography. So it seems like the spatial constraint is really important for generating this topography. And again, if you can look at these, this is a task-only model, and this is a unoptimized model. It really doesn't have any smoothness in it. It's really like salt and pepper. It doesn't have any structures. If we look at the SOMs, the DNN SOM overshoots, again, like in V1, it's too smooth as compared to the brain. If we look at Merlin Berman's and Nick Glauch's iterative topographic neural network, it does generate features that have a similar smoothness as the brain, but it actually doesn't replicate as well the categorical structure of the brain. So this is kind of really interesting because we have a very simple rule that we're doing a self-supervised learning, and our spatial constraint really just encourages nearby units to have nearby responses, and we have like one rule that generates this whole as functional and spatial topography of the ventral stream from the early visual areas all the way up to the high-level visual areas. So once we have a good model, we can ask kind of what properties of the models are really good for replicating kind of the structure. So how important is a task, and how important is our spatial loss function? Because we could come up with many training tasks, and we can come up with many spatial loss functions. So what I'm gonna show you on the left is qualitatively the TDANN spatial topography in V1 on the top and the VTC on the bottom. And I'm gonna measure the smoothness of these maps and compare it to the primate data in the green on the right to have the quantitative comparison. So I told you about this the self-supervised task and the relative spatial loss. What if we change the task? For example, a lot of people have said, including DNN, who's a co-author in this paper, that categorization is a really good task for it really replicates the function of the ventral stream. But if you look at the categorization task, the orientation maps in V1 actually don't look as good and are quantitatively less smooth than the ones that have in the TDANN. And as I've showed you before, it actually does not even replicate. This is a different one. This doesn't replicate actually the spatial structure of the brain. So even though we have a spatial loss here, it actually doesn't generate a clustered region that the self-supervised algorithm generates. If we change the training, the spatial loss to how do you call it, to an absolute spatial loss that really minimizes the physical distances in the brain, and this has been a loss function that's been used by the DiCarlo lab. We see that this loss function is too strict, and it actually makes a map, again, if you look at V1, and also in VTC on the bottom, it makes the maps much less smooth, and actually in V1, they don't replicate the features of the V1 as well as the self-organizing map. So it turns out that both the training task and the spatial loss function matter, and we think that the self-supervised task is one more biologically feasible. And second of all, maybe it just generates generally useful visual representation, and this is why it's kind of useful for the brain to kind of learn this kind of task, and this is maybe why it's replicating both the functional and spatial topography of the brain. Okay, so this is how am I doing this time? Okay, so this is the first part of the talk. So the question that we have now that we've kind of found that we have this kind of tool and it replicates really well with a single rule as a functional topography of the ventral stream, we were wondering if it can actually explain a really large-scale structure like streams, because there's a question, why are there streams in the first place, and why are there like 3 streams in the human brain? So again, these streams are functionally and anatomically determined. So the ventral stream goes from the occipital lobe to the ventral temporal cortex. The dorsal streams goes to the parietal cortex, and the lateral stream goes to the superior temporal cortex. To show you all the streams in one view, I'm going to take this view and I'm going to flatten it. So this is a flattened cortex of a right hemisphere individual. So the dorsal stream is going to be on the top. The ventral stream is going to be on the bottom, the lateral stream is in between. And here I'm going to really show you why might there be divergence at the endpoints of each of these streams. So the prevailing hypothesis in the cognitive neuroscience literature is that different streams have evolved over evolution to support different behaviors that can be done in parallel, which makes the visual system very efficient. So the ventral stream is thought to be involved in object categorization and visual recognition. The dorsal stream is thought to be involved in spatial localization, for example, detecting where objects are in the scene. And the lateral stream is a more neuro-discovered stream, and people still argue what it's doing about, but it seems the common prevailing idea that has emerged that it's maybe involved in figuring out or comprehending the action in the scene and maybe the social interactions in the scenes. But the general gist is that it's involved with some sort of dynamics. Another hypothesis, which was basically the spatial constraint hypothesis, is that the brain in general learns useful representation under the spatial constraint that favors shorter connections. So I'm going to refer to the first hypothesis, which is the predominant one, is the multiple behaviors hypothesis, and the second hypothesis is the spatial constraints hypothesis. And the cool thing is now that we have deep neural networks, we can actually pit these hypotheses next to each other by implementing each hypothesis computationally and seeing which one better explains the streams. So to explain the multiple behavioral hypothesis, we're going to train 3 different networks, each one with a different architecture, because each one is optimized for a different task. So we're going to use the ResNet to do object categorization, because that's the best network that we can do object categorization, the same network that I showed you before. We're going to use, it's called a fast-slow network that has a slow and a fast channel that can actually process dynamics to train for action recognition, which is a hypothesized function of the lateral stream. And we have a third network that's trained to detect where objects are in images without even knowing necessarily what they are. So after training each of these networks to optimize their behaviors, we're going to put them in one bin and see if we could actually have the mapping between each hypothesis behavior and the relevant stream in the brain. To test the spatial constraint hypothesis, we're going to use the same TDANN that I'm going to show you, that I've showed you before. So we basically use the self-supervised training with an intermediate loss weight, and we're going to use a relative spatial loss. And here what we're going to do is what we're going to code the units in the last simulated cortical sheet topographically. And the question is, will units that are close to each other on the cortical sheet map to the relevant stream? So visually, what would be like the predicted outcome? So we've trained the networks and we want to see the best functional match between units in the last layer and the brain. So the prediction of the multiple behavior is that units that are trained on detections will map to the dorsal stream, units trained on action recognition will map to the lateral stream, and units trained to categorization to the ventral stream. And for the spatial constraints hypothesis, we hope that clusters of units that are adjacent on the simulated cortical sheet will map to the corresponding stream, and that will have sort of a continuous topography overall. Now, note that when we're doing these comparisons, we actually have to map each unit to a voxel in the brain, which turns out that this is not the standard way that people actually compare brains to models or models to brains, because typically the move is to do some kind of linear combinations of model units to predict a neuron or a voxel in the brain. So we really wanted to develop a one-to-one mapping between a unit and a brain to understand where the best match is. So to do so, we're leveraging Kendrick Kay's natural scene data set, or the NSD. This is data from 8 subjects. Each subject saw a unique set of about 8,000 to 10,000 images, and all subjects saw a common set of 515 images. This is high resolution data, 0.8 millimeters at 7 Tesla. So what we can do right now is, after pre-training our networks, we can feed both the networks the images that the subjects saw. So now we have a matrix of images and voxels for the brain, and we have the same matrix of images by units from our TDANN. And basically what we want to do is find the best match between each model unit and the brain. So we're matching the function of these units, and we're using an iterative algorithm that minimizes this cost function based on the consequence algorithm. Once we kind of find the match, we can evaluate our network, so we can look at spatial correspondence to test the predictions that I showed you before. And then we have a set of 515 left out images, so we could actually evaluate independently how good the functional correspondence is between the network and the brain. So let me show you some results. This is the mapping of an example subjects between the multiple behaviorals model and the brain. So you can see that the units, the voxels are colored in the brain by the units, the training task of the unit that best matches it. So basically, if you see a lot of red here, that means that these units are trained on the categorization task. So you can see a couple of things. First, the units from all tasks are actually mapped to all regions. So you really don't see a lot of red in the ventral stream like you'd predict, which is surprising. And second of all, it doesn't really have a smooth spatial structure like you would expect. So this was actually mind-boggling to us. If you look in contrast to the TDANN, you can see that it actually replicates the spatial organization into streams much better, where you can see we have much more smooth maps and clustering by streams visually. Of course, we'd like to quantify this. So in order to quantify this spatial correspondent, we're going to actually evaluate how many units are going to be mapped to each stream. So the chance level is going to be 33% because we have 3 streams. We did the same kind of mapping algorithm to map each human to another human because we assumed that the best that we could do is that one human's brain will be the best model to another human's brain. So the noise ceiling is about 80% correspondence. So if you look at the multiple behaviors model, you can see that only a third of the units, it's really a chance, only a third of the units are mapped to the corresponding stream. And this is, for me, it was especially shocking for the ventral stream because so much work has shown that categorization is really good at predicting the ventral stream, but only a third of the categorization trained units were mapped to the ventral stream. The rest of them were equally mapped to the other stream. So this is really unexpected for us. If you look at the spatial constraint model, you can see that it's doing much better than chance, and it's doing much better than the multiple behaviors model. And actually in the ventral stream, even if we're doing a self-supervised task, it is actually approaching the subject-to-subject noise ceiling. So maybe the multiple behaviors is not doing a good enough spatial correspondence, but maybe it's actually better predicting the function of the neurons. So what we're going to do is we're going to measure the functional similarity, how well it predicts the responses to the 515 left out images. We're going to evaluate it for each stream separately. And then for the multiple behavioral model, we'll see how well the functional correspondence to that stream is based on the training of that task. So you can see, first of all, for the dorsal stream, the functional similarity between the model and the brain is really poor. It's really poor. And second of all, you'd expect that maybe the units that are trained on detection would be better predictors of responses in the dorsal stream, and actually they're not better. The categorization task actually does better than the detection task. And this kind of feature is actually true for all 3 streams. For some reason, the categorization task doesn't work better than the other 2 tasks. In contrast, if we looked at the functional predictivity of the unleft out images for the spatial constraints model, you can see that especially for the dorsal and the ventral stream, the functional correspondence reaches the noise ceiling, so it's doing really well at replicating the function on the left out images. We're doing less well on the lateral stream. One possibility is this is because we don't really have dynamic images in the natural scene data sets, because the natural scene data set only has stationary images. So this is kind of cool that the same principle also explains stream, but one question is, there's this whole literature that actually shows that different streams have different functions. Do units that map, even under the self-supervised tasks, to different streams, do develop different features that might be useful for different streams. So to test this hypothesis, we looked at the transfer performance of new tasks. So basically, after training this in a self-supervised way, we built like a last layer of a classifier to see how well the outputs of the unit string mapped to the dorsal string or to the ventral string could do other tasks. So we picked a position task because that's a hypothesis task of the dorsal string, and we picked a categorization task because that's the hypothesis function of the ventral string. So if we looked at how well the units perform in this transfer task, you can see that the green units that were mapped to the dorsal string do better on the position task than the red units that were mapped to the ventral string. And in contrast, the red units that were mapped to the ventral string do better at the categorization than the green units that were mapped to the dorsal string. So even though we are training on one task and we have one spatial constraint, it is a case that units mapped to different strings do develop different features that are in line with the hypothesized behaviors of each string. So to end, our findings suggest that unsupervised learning under spatial constraint to minimize wiring lengths can explain both the functional and spatial organization of the visual system in many scales. And I find it really interesting that you have a single local spatial constraint and you can percolate across the system and even generate this large scale organization like processing streams. And on top of it, model units assigned to different streams do develop different functional properties that are still in line with the stream hypothesized function. So I'd like to kind of suggest that it's important for us as cognitive neuroscientists not only to map the function of the brain, but also the topography of the brain, because it can give us new insights about underlying mechanism. And one of the insights from our work is that spatial constraints really play a big role in shaping the visual system. So because maps are prevalent across the brains, there are maps in auditory systems, there's maps of the human bodies, there might be even more cognitive maps in higher order area. It would be really interesting in my mind to look at other aspects of the neural and functional topography to test if these principles apply to other cortical systems beyond the visual system. So I'd like to end here. So thank you so much for your attention, and I'm happy to take your